Done, sir. Today we have a very eminent speaker, Dr. Pankaj Kaul. He is the Dean in Rahat Behera University, Mohali. Earlier, he was in the PGI, Chandigarh. So I request Dr. Pankaj Kaul to please start his presentation.
Thank you so much, sir, for a brief introduction. My topic is going to be on role of medical lab science in healthcare industry and career opportunities in it. As you have already introduced, I am Dr. Pankaj. I have done my master's in medical lab technology with PhD in India from PGI itself. And then I had a fellowship from UK. I'm right now presently working as Dean in University School of Allied Health Sciences, Riyadh Bahra University, Mohali, Punjab, Bharat. I also belong to a very big association, which is Indian Confederation of Medical Lab Science. I am the general secretary of that association, and this is the logo of my institute where I am serving. But this is not my right introduction. My actual introduction is that I am a voluntary blood donor and a centurion blood donor with more than 114 blood donations up my sleeves. I have also donated my eyes, my body, and my organs. That is my exact introduction, what I am for. So, coming on to the topic, what makes one type of cell different from another cell type? That depends upon the chemistry of the cell, its vitamin composition, water, enzymes, composition of amino acids, mineral and proteins, RNA, etc. How these atoms are put together in the cell determines what the cell looks like, what job the cell can do in the body, and how the cell communicates with other cell in the body. Since each type of cell has its own particular chemistry, each disease too has its own particular chemistry. Similarly, in the clinical laboratory, each section plays its own role in its own way. Like in clinical lab, microbiology lab, we identify the organisms, we do biochemical testing, then we determine the antibiotic sensitivity, which ultimately helps the patient treatment. Similarly, in virology lab, tests are done on the serum for antibodies directed against viruses such as HIV, COVID, hepatitis, etc. in a very sterilized conditions. In the blood bank, we screen for infection and determine which antibodies are present in plasma so that a compatible cross match
Dr.
can be
Pankaj,
done. Yes, sir.
are you moving your PPT or not?
Yes, sir, I'm moving.
But it is not moving on the screen.
It's not moving on the screen.
No. Please test once more. Test it.
Okay, should I start from the beginning then?
Yes, just you go through the PPT.
Okay.
Go through the PPT till you have
moved.
Yeah. Just try to move. Yes, you try to move it.
Is it moving now?
No, not yet. Can you enlarge it and see what happens?
I'm in full screen itself, sir.
But here only on the side I can see small PPTs.
It is only on coming on the side.
It's not coming on the side. I cannot see. I am seeing the first one only. Only first one. Uh, yes, yes. Now, okay.
Is it okay now?
I can see the title already. The title one.
I'll change the next.
Yes, please.
Online.
No, the same one is there. The title PPT.
Is it now okay?
Yeah, yes.
Is it
Yes.
coming now?
The first one has come now. Second one.
Okay.
Third, chemistry. Chemistry. Yes.
Yeah. It is okay now, probably.
It's okay.
Okay. Okay. Actually, the moment I make it at full screen, it is not showing. Now I am doing from the small screen. Probably you will accommodate me.
Yes.
So I'll start it again, sir.
All right, please.
What makes one type of cell different from another? It depends upon the chemistry of the cell, which has got vitamins, water, DNA, enzyme. It is the chemistry of all these things, protein, RNA, etc. And how these atoms are put together in the cell, that determines what the cell looks like, what jobs the cell can do in the body, and how the cell communicates with other cells in the body. Since each type of cell has its own particular chemistry, each disease too has its own particular chemistry. Will you please unmute? Someone is interrupting in between.
Yes. Please unmute your mics. Unmute. Please mute your mics. Mute your mics.
Since each type of cell has its own particular chemistry, each disease too has its own particular chemistry. Similarly, in the clinical laboratory, each section plays its own role in its own way. Like while we are working in a clinical microbiology, we identify the organisms first, which are collected from the patients. We do biochemical investigations to identify what type of organism is it, and then we determine its antibiotic sensitivity, which ultimately helps in the patient's treatment. Similarly, in virology lab, we test the serum for antibodies, which is directed against viruses such as HIV, COVID, hepatitis, etc. In the blood bank, we screen for infection and determine which antibodies are present in plasma so that a compatible cross match can be done for safe blood transfusion. This makes a clinical lab professional. A laboratory professional is a person who performs laboratory testing on blood, urine, feces, spinal fluid, and other body fluids in addition to cancerous tissues. They provide more than 70 to 80% of the objective data used to make clinical decisions. Laboratory professionals conduct tests to verify a potential dangerous drug levels, detect a cancerous tumor with DNA techniques, identify toxic agents such as anthrax in the cases of bioterrorism, monitor the level of anti-rejection drugs in transplant patients, type and cross-match blood and organ samples for transfusions and transplants, identify the causative microorganism in a blood or wound infection. Such vast is the area which we are working in. Various synonyms of these laboratory professionals are like biomedical lab scientist, clinical laboratory technologist, medical lab technologist, clinical biochemist, cytotechnologist, histotechnologist, phlebotomist, and many more names are there, but there is no system all over the country. What makes an eligible medical lab technologist is that a person with a doctorate master's, a bachelor degree, and certification as a clinical laboratory scientist or medical lab technologist MLT, capable of running lab independently or supervise a clinical lab work independently. Then comes, what does a phlebotomist mean? A person who has been trained to draw blood for laboratory tests. A phlebotomist goes through a training program and must have knowledge of all types of anticoagulants which are used during the investigation. Pre-analytical things, I mean to say. They can do so on the job training also in the hospital. And a histotechnologist is a person who prepares solid tissues such as biopsy samples of tumors for examination under the microscope. A person doing so with specialized degree in histotechnology is a histotechnologist. And a cytotechnologist prepares and examines slides of exfoliate cells. They are the best known for the performance of Pap tests for cervical cancer, but they examine many other types of specimen as well. A person with a specialized degree and or certification as a cytotechnologist. As far as the history of medical lab profession is concerned, the professional requirement of medical lab science advanced from no formal qualification in 1940s and 1950s to diploma, bachelor's degree, followed by master's degree and doctoral degree program in the last 30 or 40 years. By the 1980s, many degree granting institutions established their curricula in medical lab science and have thereafter mushroomed throughout the country and globe like anything. Today, medical lab science is a well-developed profession of knowledge that includes portions of basic and medical science, medical lab techniques, and research methods. This facilitates and ensures the production of quality medical lab diagnostic testing serving mankind. As far as structure of lab service is concerned, a medical laboratory service must be integrated with the healthcare system which exists in the country. This must function as a network which could be accessible and provide a service that is needed by the community, like we have faced in COVID and other abrupt epidemics. This networking must consist of a community-based healthcare center, which includes comprehensive health center labs, investigation, controlling, and preventing major diseases in a community, staffed by a qualified lab personnel who promotes diagnostic health education and awareness and is able to perform basic tests. There should also be availability of maternity health care units staffed with trained nursing staff supported by a qualified medical lab science personnel who can perform screening of anemia, proteinuria, sugar, etc., collecting blood for antibody screening or for examination of parasites. District hospital laboratories, in addition to the work which I have stated above, the district laboratories have an important role to play. They must supervise the work of community-based health center labs and testing referred specimen which comes from there. Must have facilities for clinical investigation and epidemiological surveys. Specimen collection and transportation system should be there, and patient attending health centers must be benefited from the facilities of district health labs. Epidemics to be investigated rapidly at this level. Then comes the regional hospital laboratories. In addition to the work stated in the above said labs, it must assist and supervise the district health labs, analyze referred samples, perform a range of specialized tests, investigate epidemic and do more clinical and public health facilities, train lab technologists by organizing refresher courses or continuing medical education programs. Then comes central or national healthcare laboratories. In addition to the work stated above, these laboratories must assist and supervise the regional health labs, analyze referred samples from DHL and RHL, perform a range of super specialized tests, investigate epidemics, and provide more clinical and public health facilities. Must train lab technologists by organizing refresher courses and conferences for the C group of people. And these centers must try to accredited themselves with the WHO. This is the tree of this laboratory service network, like primary, district, regional, and central reference laboratories. Then comes the professional ethics in lab science or lab medicine. Earlier, the name, which was laboratory technology, it turned out to be lab medicine, and now it has been start calling as a lab for medical lab technology. It became a lab sciences, and now it has been called as lab medicine also. It is a branch of moral science which is concerned with the obligations which a member of a profession owes to the public, to his profession, and to his client. Such are the professional ethics we must follow, and we must consider this lab medicine in truth is a profession, not just a job. And as a profession, it makes certain demands on its members related to the maintenance of certain professional standards of behavior, conduct, attitude, and work, all aim to give the best service to the patient who are served by this medical team. Once we put on an apron, that apron doesn't mean just a white coat to prevent yourself from the infections we are handling the samples. It has got a lot many meanings. We have to show that behavior and conduct and attitude while we are putting on the apron. As far as objectives of this profession is concerned, that there are few main objectives as far as professional code of ethics is concerned. There must be professional privileges, behaviors, and responsibilities towards the member of the community, to promote professional quality, to defend private professions from undue interference by the government or by other agencies. Here I will say we are right now struggling with the Ministry of Health for making a model curriculum. We started work on this since 2012. Only after 3-4 years we could get a model curriculum in 2015-16, and now again a revised model curriculum is coming up. Earlier there was no this revamping of this curriculum for 30-40 years. Now within 5 years they have started remodeling it. So such is the interference. So for this we have tried our level best to enact the Allied Health Science Council, and we are working on that. And very shortly, this council matter will come up, and we will have everything in a streamlined manner. To preserve the dignity of profession and the confidence of the public, and further to defend clients from unscrupulous individuals. You know, you will be surprised to know how we are to defend the clients. People are doing taking undue advantage of our very pious profession. Only symptomatic treatment is being given by the clinicians, whereas they are taking the investigation, they are giving an investigation list to the patient, and they are only ticking few. It is an indication to the lab concerned to only do those tests which are been ticked, but those labs are charging for all these investigations from the people. So that is making a very bad reputation to our lab profession. So we must defend the clients from unscrupulous individuals who are doing such malpractices. We must fix certain standards of the compensation for service or work. As far as our responsibilities is concerned, once we become a medical technologist, we must accept the responsibilities inherent to being a professional. The logistics of this profession is, as I enter the practice of the medical technology, I shall uphold the law and shall not participate in illegal work. Act in the spirit of fairness to all and the spirit of brotherhood towards other members of the profession. We must accept employment from more than one employer only when there is no conflict of interest. There are so many people who are serving in big institutes, but they are doing their private practice also. They are putting in more efforts in their private practice than they are working in their government jobs. So we must perform our task with full confidence absolute reliability, and accuracy. We must share our knowledge and expertise with our colleagues. Will probably someone is interrupting? Please, please mute the speakers. We must contribute to the advancement of the professional organization and other allied health organizations. We must restrict my praises, criticism, views, and opinions within constructive limits. Our profession is such a pious profession where everybody gets experience of life from its work. And from that, we must try to share that experience with others. If we will retain that experience with us, then it will be very difficult for our growth. So we must try to share our knowledge, whatever we get. We must restrict the criticism. It should be a positive criticism. Only then we can advance in our profession. We should be confident. We are not to leak the secret of a patient as far as our lab investigations are concerned and results are concerned. We must be very confidential for the patients. We must work with full dedication, uphold the dignity and respect of our profession, and conduct a reputation of reliability, honesty, and integrity. We must be dedicated to the use of clinical laboratory science to promote life and benefit of mankind. We must also be very much vigilant about the violations, if anyone, and to the principle I hereby subscribe and pledge to conduct myself to all times in manner befitting the dignity of my profession. As far as National Skill Development Corporation is concerned, as per the Ministry of Health and Family Welfare, New Delhi, latest these reports have said that National Skill Development under Health Department projected at least employment base of, we have right now employment base of 35.9 lakh at present in allied health sector against 50 lakh required. So still there is a deficiency of trained professionals in this field. And the study thus indicates that there is an acute shortage of allied health professionals in the country. Employment of laboratory technologists is projected to grow 5% from 2022 to 2032. It is faster than the average for all occupations which are existing at present. About 24,000 openings for clinical laboratory technologists are projected each year on an average over the decade. There are various career options of medical lab technology in the healthcare industry. This field is endlessly variable and is ever growing. With lot of diversities, new technology emerging online daily, responding to challenges in caliber and competence of work standard, therefore needs regular updating to remain in pace. There was a time when we were having only MiGs and jets, but now we have to prepare ourselves for Rafale also. So in the same way, we had to keep our knowledge at pace with the growing technologies. Various career options are available in medical lab technology. As medical lab technologist, biomedical scientists, there are so many opportunities available in hospitals, research institutes, defense, railways, non-government organization, and charity health centers. As research assistants in universities, government institutions, health organizations, pharmaceutical industries. As an analyst, there's so many diversing field in food and beverage industries as quality control officers, food laboratories, in teaching as professors, lecturers, and vocational teachers. In schools also, they have started these vocational courses. So there is a breakthrough there also. As a medical representative in pharmaceuticals, humans, as well as veterinary. Once you do a bachelor's, nobody can stop you to appear in Indian administrative examinations, civil services, and foreign services. You can go there and work in national government services. If you want to upgrade your education or qualification, you can go in for post graduations like Masters in Medical Lab Technology, or now it has become Medical Lab Sciences. Microbiology with specialization in parasitology, virology, mycology, bacteriology. Biochemistry, you can do clinical biochemistry, hematology, histopathology, immunopathology, cytology, and gynec pathology, pharmacology. There are so many fields available. We can also join Master in Science in Basic Sciences also. So since we were doing all these things, so much super specialization was there. The career of a technologist was getting very much focused. Keeping this into our consideration, since I was at the task force committee of the 4-year curriculum, model curriculum, which we have made this as a master's in pathology with specialization in so and so. Like we are having MD pathology, like that we have made this master's in pathology and likewise. So once we have this education and basics, we must have dexterity in action also. There was a problem once in our this tissue processor. It was not doing proper vacuum suction. It was an imported instrument. So there was just a petty things, just putting a strip in the lock. I relocked it and it started working functional. Because it was imported and the engineer was to arrive from New Delhi, it was very difficult for me to wait. And because this process is at least 250 biopsy samples once in a time. Otherwise, it was very difficult to cope up with the work of PGI. So this was the immediate work I could do. I informed the engineer that I had done. He was very happy, and thereafter when he came, all things were set right, but my work did not stop. Then there was a correcting of hazardous vent pipe of autoimmunostainer. This was also an imported machine. There was a little problem in this. There was a small plastic junction which was to be imported. It would have taken months together. But I took a small piece of that IV set. There is a junction in that also. I fixed it here and it started working. Once there was a shortage of soap, and during rainy season, soap gets very soft. It becomes soft and it gets waste. So this is the indigenously made strip. On that, a magnetic catcher was kept, and I used this cold drink lid on this, and I halved the soaps so that all the soap doesn't get wet. So it is only half of the soap was used, and it used to get dried because there was no contact with the floor or soap case. So it worked for longer, and we could evade the time of shortage in our institute. So this is how you can help your institutes by innovations. But that is possible only once if you have got an innovative research oriented mind and if you have got innovations in your mind. Then there was a load of samples. Too many samples were coming. We had only one gross station, but this was a temporary gross station with this silicone base on this. We fixed the simple kitchen shank. So we converted into a gross station. These are the ideas. This is a rotary sort of sharpener machine whose ground glass was broken from one side. It was not of use. Just by making a small wooden stand, I could make a small teapot out of it. The scissors was broken. I just took a pen, this ball pen refill and fixed it here, and it is still working. You will be surprised to know. And one more wonderful thing I would like to share, that we had a air condition engineer. His bag used to tear every fortnight or 1 month because of these tools and all that. So I gave him an idea to use this 10-liter this plastic container. So it was cut transversely and made into two parts, and he used to put all these tools in this container, and then he used to put this in bag, and the life of the bag survived for more time. So the moral of the story is this: once we have 3 Ds, desire to join this profession, then put on our dedication, and with our dexterity and professional ethics, we can do wonders in this lab profession. Thank you so much for listening patiently. Thanks, sir.
Thank you very much, Dr. Pankaj Kaul. It was an excellent lecture. We came to know so much about medical lab technology. There are many things we did not know, and today we know about MLT. And I was very impressed when you said that I'm a regular blood donor. You have given more than 100 times blood. And not only that, you have donated your whole body for research. Very, very few people do this. This shows your dedication, like you said, the 3Ds: desire, dedication. And that's really very noble of you that you have donated your body for research after you go away. Should follow this example. I'm very impressed by your, thinking. Now, the House can ask question to Dr. Pankaj on MLT and the career opportunities. Do you have any questions for Dr. Pankaj? You can ask him. I think they have understood very well.
Hello, sir.
So, yes.
Sir, myself, Batula Narsimlu, sir.
Yes.
I am from NDRA Karnal.
Yes.
Sir, I want to interact with sir.
Please.
Sir, go ahead.
Sir, during this laboratory practice, is every protocol is following GLP or whether normal ISO?
Sir, it depends from person to person. Since unless this our council doesn't come into picture, things are going like that. Everybody is doing his own protocol. There is no standard formats yet. Once it comes into picture, then everything will work in a proper way. There was a problem after enactment of this Allied Health Profession Council. The government is going very sluggish. We had been waiting since 21. They didn't do any work. We have put up a case as a public interest litigation that why don't you execute this enactment? So we are waiting for that. Once it comes into picture, things will come in a proper way.
Okay, thank you, sir. Because you shown a single example now, sir, that soap how we can keep in rainy season. So if is there any protocol for it or it is new innovative? So that's what I asked, sir.
It is just my own ideology. I am just doing these innovation just for the as I'm having a sort of innovative mind. I do some or the other thing. I keep on doing. I play with my plants also. I've got a terrace garden. I keep on playing such games. And in health sector also, I am doing a lot of things. So these were just an example. I had also presented these pictures in international conference, and it was appreciated like anything. We all must try to come out best out of waste.
Thank you, sir. Thank you, sir.
Dr. Prakash, are you there?
Yes, sir. Good evening.
Good evening. Please ask him question.
Yes, I don't have any question rather than complimenting the speaker for this wonderful lecture, but just I wanted to add is about our Phenome India cohort project of CSIR, which basically we are looking for a cardiometabolic disorder, and where we actually are looking for this people with the MLT, medical laboratory practice, for collecting the blood sample, then performing different tests that are actually required in this project. So this is actually All India cohort project for 5 years, where IGIB New Delhi is coordinating, and we are actually part of this project. So just as sir was telling the scope in MLT and the requirement of the trained manpower. So from that angle, I just thought I will just add from our side.
Very good.
Otherwise, it was a wonderful lecture and lot to learn actually from this lecture.
Yes.
Thank you very much, sir.
Sir, in that, if you will just keep in touch with me, I can provide you good people for this, and we can also have a collaboration with our university, and we can provide you manpower for collecting samples and all that. We can have a MOU type. We'll be there for the service at any time, sir.
Very nice.
Definitely,
Dr.
I will
Sunil?
talk with this the people, those who are coordinating in IGIB, because we are actually looking for a very big data set and for a longer duration.
Right,
So
sir.
Dr. Santanu Sengupta is the PI of this project at CSIR level, and all the 37 laboratories of CSIR along with our headquarter actually is involved in this. So at least 20 to 30,000 is actually sample we are going to collect in the initial phase, and then that goes to in the 3 different phases, looking for the different tests, and this actually our volunteers will be the employees as well as the retirees present and their spouses. So 5 years course study and very extensive actually information we are actually expecting from this project. Thank you.
No problem.
Thank
sir.
you.
Anytime, any
Sure.
service is required, we're
Definitely.
most welcome, sir. You're most
Definitely.
welcome. Thank you, sir.
Dr.
So nice.
Pankaj,
Yes, sir.
I'll be requesting you very soon of MBSI. I hope you'll accept it.
Sure, sir. Sure. Definitely.
I'll be very happy. And now, Dr. Sunil, can you please discuss something?
Yes, sir. I just want to add up. Sir is already doing very good for the, nobody is doing it. Sir is doing like you. Like means sir is working for the students and for forming the like council for the medical lab technology students. Nobody has done it, previously. So sir is doing, a novel work. If I will be the president, I can give you, sir, a very good award, like, something very big. But because sir is doing, which is not possible even, right? Because in MLT, what happens, all the people, they meets unity, lacking of unity is there. People is not supporting, people think, okay, we are from technical background, we are technicians. So these kind of things are there, but still sir is, I don't know, sir have some natural powers or supernatural powers sir is doing. Without alone, sir is going out of the box, and sir is like a very good leader. I just want to add these things only. Sir is very helping and very nice person. I have came across like you, sir. Sir is very humble and helpful for the students, for the staff, for the people. Sir is always, always there, 24/7. Sir will never,
Thank you so
ignore
much.
anyone.
Thank you so much. Yes, thank
Yes.
you.
So, sir
He's
is,
my neighbor. He's my neighbor.
I don't have
I know him very well.
expressed these things.
Okay, anybody
Yes,
else
sir.
want to say something?
So, sir,
Anybody?
sir already know, because nobody can judge sir about any topic. So,
Hello.
sir explained everything very nicely.
Thank you.
Hello,
Thank you.
sir.
Yes, please.
Hello, I'm Mayura from Yevlanasi. Hello, can you listen me?
Namaskar,
Yes.
ma'am.
I was just curious to know, means I don't know the depth of this field. Actually, I'm a MSc microbiology basically. Is the government gives some support for starting up some new labs, like private labs? They do give some funding.
I can't listen. There's no sound. I can't listen you, ma'am.
Hello,
Yes.
you listen me now?
Yeah,
Yes.
it's okay now.
I was curious to know if somebody wants to start a new setup in this medical laboratory field. Is government is providing some funding to like some startup new lab?
No, madam. These things are available in banks only. Government as such is not providing.
Okay, so means
funding and all that.
SMR or
Only,
CSIR or
only if you are in a government service, and
Huh.
and especially when you have got a MBBS degree or MD degree, only then they will give you some project. Or if you are a faculty of some government institution, they are giving you projects also.
That I am aware,
But
but
not
for
not like that you want to
no,
start your own private lab and government will fund you. No such
like
things are available.
no collaboration they do with the government lab.
With government labs they will do, but if
But
you
not for
want
the
to set
private
up a
lab.
private, no, not at all.
Okay, that is
I am
not.
not fund, but I had some project with the UK
Uh-huh.
Institute of Biomedical Lab Science, UK. I had availed that global sort of funding from them on a competitive basis. I submitted my project and they selected my project and they gave me a funding of more than $100 or something like that. They had been giving, but India didn't give me any funding because I was not a faculty. That is why they refused to give me anything.
Okay, so in foreign countries, you can think about the
Yes,
fundings if you have some
yes,
competent project with
yes,
you.
yes, yes. They
Yeah,
are
thanks for the
providing some
information.
incentives, but in India you don't get.
Okay,
This is
thanks for the
unfortunate.
information.
This is
Okay,
unfortunate.
Dr. Malini, you have some question?
Yes, sir. Yes, sir. Yeah. Good evening,
Yes. Good
everyone.
evening.
I have one quick question. I am from a rural area college, and it is undergraduate college. We have microbiology honors. So I was asking, sir, like the students who are freshers, or even if some goes for the MSc, like how can they avail this sort of opportunity? Because in West Bengal, like in government sponsored institutions, these courses are not in operation. Maybe some private institutions have started or something of that sort. So how can the students get this footfalls in this area?
Ma'am, I'll just tell you a story. When I was struggling for my existence in this profession, I started my career from a lab assistant. I was working on this intestinal parasites. Some Ayurvedic drug was being used for intestinal parasites in Himachal Pradesh, Joginder Nagar Regional Research Center, Ayurveda. That was my first exposure to this field. I was to work on these stool samples. I was making that saturated saline and I was just doing flotation method. I was looking for these parasites. I was surprised. I was amazed to see a totally wonderful world in our stool. That really made me so curious about this subject. So I looked for where I can do this course. At that time, I had already done my BSc. Then suddenly, after a few months, I came across an advertisement of PGI where I came to know that there is a medical technology course being run in PGI. I came over, I prepared myself, I appeared in entrance examination, I qualified, I did BSc. I was told at that time by the counselors, by head of the Department of Microbiology, Professor K.C. Agarwal was there. Told, call you, come tomorrow, I'll give you a job. I told, no, sir, I want to know the funda of this field. So I did 2 years BSc at that time. I was not satisfied. I still prepared for all entrance examination are there in PGI. We are not getting just by chance. We had to appear in entrance examination. In MSc, I qualified. I did my MSc microbiology with parasitology as special subject. I didn't leave that parasite. I didn't leave. Then after some years, I got more inquisitive about learning more. I appeared for PhD entrance examination. My guide was Professor N.K. Ganguly. He was DG ICMR thereafter. And the chairman of my doctoral committee was Professor R.C. Mahajan. He was the Secretary General of Indian National Science Academy. I have had a very tough time to learn, but main problem is that I didn't get any incentive all through this. I just kept on satisfying my egos. I must learn more. I must learn more. And ultimately, I reached to this extent that now I had no other way out to promote this profession than to come as a dean in an institute. And I'm trying to make it an institute of excellence. And I'm trying to create these budding students, budding technocrats, making awareness. I'm going to, from school to school, giving awareness about this field. Because the day I joined PGI for this medical laboratory technology, you will laugh on me. I was discouraged like anything. My parents were told by the people who came to know that I got selected in this field that what he is going to do in PGI, he will just test the stool and urine of people. So it was a very great setback to me. Then there was a Ramakrishna Mission ashram here in Chandigarh. I went to one Swamiji, he was very well known to me and I was very fond of him. I told Swamiji, what is all this rubbish? I may have to go back. He told, no, beta, you just tell these people that if this urine and stool will stop, you will definitely come to me. And that gave me a sense of inspiration. And I started my work in this, and I have reached to this extent that it was only medical technologists who saved the people in COVID also. In dengue epidemics, it was a technologist who saved. And there was one engineer, I went once to see this Bhakra Dam. There was an engineer who asked my children that, beta, can India make another such a powerhouse? Because it has got so much of cement has been put on this Bhakra Dam that at 3 feet road can be made around the globe. So much of cement has been used. Do you think? My children told, no, uncle, so much of money has been spent on this. He told, no, you can make another Bhakra Dam. How? He told, just switch off electricity when it is not used. The thing you are saving, it means you are generating that thing. In the same way, a technologist is doing the same work. If we give a proper diagnosis in hospital, like PGI, it's having so much investment in PGI and All India Institute, that a patient who was living for 1 month for getting his treatment, if we give a proper diagnosis at a proper time with minimum turnaround time, the patient will be discharged within few days or within a week. So in the same time, we can put on 4 times patient in the same that capacity. This is the beauty of a technologist. We can save the patient from long hospitalization. We can save the expenditure of government on that patient. We can save the time of the clinician, everything. So I love my profession like anything, and I want to spread this message throughout the world.
Very good, very good. Thank you very much, Dr. Pankaj. And it was a very inspiring talk and discussion.
Thank you, sir.
We were really enlightened by your words, by your talk. Thank you so much.
Thank you, sir.
Okay, we can conclude the session now.
Thank you, sir. Thank you so much. Thanks for giving me a chance to spread my message around the world. Thank you, sir.
Thank you so much.